So we will start with simple diffusion. So simple diffusion is spontaneous, passive movement of the particles they call solute from area of the high concentration to the area of the low concentration, or from region of the high temperature to the region of the low temperature. So in here we will see again, so this area, this green, some kind of particles in the water, and this is area of the high concentration, and the particles will always start to move over time and to the area of the low concentration. So this process called diffusion, and it's called passive transport because it does not require energy on ATP molecule in order for these particles to move. And in here we have example, the drop of the ink. Yes, so if this is green all particles in here, this is drop of the ink. When we place ink in the water, they dissolve and they will evenly distributed along all this is area. They will move from from area of the high concentration to area of the low concentration. And again, simple diffusion, it's gases moving in and out of the cell. If we have on one, in this area, on this side of the membrane, yes, so this is the membrane. And if we have in here high concentration of the oxygen compared to this side, inside of the cell, for example, low concentration of the oxygen. So oxygen molecules will move, diffuse through this is phospholipid bilayer or membrane inside. Same about carbon dioxide. It will move from the high concentration to the low concentration. And in here show one of the examples. So this is a bead, and if we will put bead in the tube in here, in the water, and over time we see this is after 15 minutes, what happening after 15 minutes. So we can see that from this bead, this is pigment, this is one cell of the bead showing in here. And inside we have big droplet of the pigment. So when we cut the piece of the bead, this we rupture this cell, and pigment, of course, will leave the cell. And in this case, this pigment leaving these cells in the bead, and over time, first it's a clean water when we just put the piece, but after 15 minutes it become pink. After 30 minutes, it's become even more reddish. After 45 minutes, it's very dark become. So this is diffusion of these pigment particles from the area where they have high concentration of the pigment inside of this piece to the area of the low concentration in the water. And with time, we will see that more and more pigment leaving the piece of the bead. So on this image show difference between this process diffusion of these particles of the pigment also depend on the temperature. So if we have 0 Celsius, so 24 Celsius, 50 Celsius, 70 Celsius, this is temperature of the water. And we put same amount of the water, we put same piece of the bead, but if we have water with different temperature, and for example, after 5 minutes, what we will see? So after 5 minutes in this different temperature, we will see different speed of the diffusion occur. After 5 minutes with 0, almost no pigment diffuse. In the 20, we see more diffusion occur. It's become more pink. In the 50 Celsius temperature of the water, even more, we have much more diffusion. It's become redder. in the 70, even more happening. So we have more diffusion happening. So the higher temperature, the faster diffusion will happening. This is what it's showing here. So the speed of the diffusion depends on the temperature. The higher temperature, the faster this diffusion will happening. Okay, so passive transport. So why it's important? Yes, so to create energy, for example, we will use cell will use passive transport to help exchange gases. For example, oxygen, carbon dioxide. It's exchanging during respiration, photosynthesis, transpiration of the plants, exhalation of the water vapors through the stomata or pores. So all this example of the simple diffusions. And then here shows that this is the cell. This is yellow in here, everything. This is inside of the cell. Inside of the cell. This is outside of the cell. And if we have outside of the cell a lot of these blue molecules, for example, it's oxygen, what will happen? Simple diffusion. And oxygen will be able to penetrate inside of the cell. Carbon dioxide, opposite, will be more concentrated inside of the cell, so it will move in the opposite direction away. So in this oxygen will continue to move inside of the cell. So this is show over time what happening, more and more oxygen penetrating until equilibrium will be. Same concentration of the oxygen outside of the cell, same concentration equal to the concentration of the oxygen inside of the cell. And you can see, if we count all these dots, will be same number of the oxygen molecule outside of the cell compared to the inside of the cell. So equilibrium achieved. Next, we will talk about facilitated diffusion. So again, facilitated diffusion is a passive transport. So it means it's moving, molecules will move from the area of the high concentration to the area of the low concentration. But it's called facilitated because these molecules, for example, outside of the cell, potassium, this is molecules, they cannot simply diffuse through this cellular membrane by layer of the phospholipids. So because of this, they need facilitated diffusion. They need this transmembrane protein. And here purple show transmembrane protein, and potassium can move through these channel proteins inside of the cell. So in this case, we have high concentration of the potassium inside of the cell, and we have low concentration of the potassium, low concentration inside of the cell. So because of this, potassium will move passively from the area of the high concentration from outside of the cell through the transmembrane proteins into the area inside of the cells where is low concentration of the potassium. Glucose molecules, and here they show red, same stuff. They also use transmembrane protein, and in this case, it will be carrier proteins. So facilitated diffusion achieved through the two types of the transmembrane proteins, channel proteins, like for potassium, and carrier proteins, like for the glucose. So how you can see in this case, this protein, it's not like carrier protein, not always open. It's locked in here. So glucose first need to move inside when protein like open like this, and it will fit precisely in here. This shape for here, what it's done, it's only for the glucose molecule. The other molecules, this different shape will not able to fit in here. So glucose will move from outside, whereas we have again high concentration of the glucose, and it will move inside of these carrier proteins. Then carrier proteins will change shape, or how to say, it change conformation. And in this case, now this glucose will be detached and move inside of the cell, whereas concentration of the glucose is low compared to the outside of the cell. So the first type of facilitated diffusion is a channel diffusion, in which involves transmembrane protein channels letting molecules through the membrane. It's again as in the example of the potassium. And second type is facilitated diffusion, but through the special transmembrane protein carriers, which can change shape, conformation, to let passing molecules through. And again, conformation means changing the shape. Transmembrane protein carriers, they change in conformation, they change in shape. Facilitated diffusion need a special proteins, channels, or carrier, which will help substances, for example, potassium and glucose, to move in or out of the cell. Facilitated diffusion is the process of spontaneous or passive transport of the molecules, ions across the cellular membrane via specific transmembrane protein or channel. So why is it called specific? Because each transmembrane channel or transmembrane carrier protein, they all designed only for specific molecules. So this is why they call like that. They need specific. And in here, we can see example of the facilitated diffusion is potassium ions, what I already showed, and also glucose and amino acids also move like this with help of the proteins carriers. And in here on the image show glucose molecule in here, and it's moving from the area of the high concentration of the glucose to the area of the low concentration of the glucose. And it's moving, this glucose moving through this specialized transmembrane proteins carrier, which can change the shape and allow glucose to move in the cell.